Okay, good morning everyone. And
Good
today,
morning, sir.
good morning. And today we are going to deal with a new topic, right? And in your syllabus, you have got a small topic called as fitting monomers. Okay, and that is what we are going to talk and do stuff about these things. Okay, so this topic, I think it has got very less theoretical implication to know you about. Most of the stuff that this topic is supposed to be dealt with is in practical aspect. Okay, so now what our objective would be is that I would kind of demonstrate you on how you can end up building a functionally active multimeric state of a protein if you have provided with the monomer. Okay, so that is what this topic is all about. How do you fit monomers around each other so as to generate a functionally active multimeric state of your protein? Okay, so that is what we are going to do. Okay, so that would be our first objective. The first objective of today's discussion would be to build a full tetramer from a pdb file containing only one monomer or only one subunit of a protein, and we are going to do it using or you can say we are going to build up or we are going to fit this monomer by applying non-crystallographic symmetries. Okay, and after we end up building a functionally active multimeric protein, then our next objective would be to study the interaction in the subunit interface. Okay, in this case, I will show you how do we do it using an example of lactate dehydrogenase. Okay, so in order to fit the monomers into each other to generate a functionally active multimeric protein, the example protein that I am going to use will be that of lactate dehydrogenase. Okay, so in today's session, we will learn to study models that contain more than one protein chain, right? Because since we are dealing with multiple chains, there would be one proteins in our scenario now. Okay, so for this, let me show you what protein we are going to work on. Okay, I I hope things are clear now. Okay, hello.
Yes, sir.
Okay, so that is what
Yes,
we are
sir. Yes,
going
sir.
to do. Okay, so now I am presenting my screen so that we can start out working. Okay, is my desktop visible to you guys?
Yes, sir.
Yes, sir.
Okay, so let us begin with our software that we are going to use. Of course, we are going to use this Swiss PDB viewer, which is one of my favorite software to do this structural stuff. And now let me locate where are our pdb files that we are going to need today. Okay, this is what we are going to deal with. Okay, so here is what we are going to do. Okay, the file that we are going to use for today's study is with pdb identification code 1ldm. Okay, so you can download it from pdb. Of course, I'm going to share this file with you on your drive as well. Okay, So this is what we are going to do. Okay, so this is the monomer of your enzyme lactate dehydrogenase, right? So well, let us first examine this protein molecule, and there are some mistakes that it might appear to you that there are some problematic things in your model if you look it at the very first instance. Okay, so let me tell you कि इसमें गड़बड़ क्या रह सकती है. Okay, so the very first thing that you might have noticed is that there are some 6 amino acid residues on its N terminus, as you can see over here, right? 6 से ज़्यादा भी हो सकता है ये, right? So there are say some 13 or 14 amino acid residues which I have just shown you. These are the ones. Okay, so these N-terminal amino acid residues are out in a solvent, right? And they are not properly folded around the protein. Okay, दिख रहा है? This much region of your protein is quite compactly folded. Okay, while the N-terminal residues appear to be out in the solvent. Okay, so proteins are not supposed to be folded up like this. Okay, proteins are supposed to be globular and they are supposed to be tightly folded, right? Okay, now one more thing that I would like to show you is that I would color the side chains. Okay, and I would color the side chains by type, right? Okay, and maybe I would color the backbone by any color. Well, say for example black would be nice. Okay, color the backbone by black. Okay, now as you can see over here, the black backbone is not currently visible. Okay, so maybe I will color it by blue on this color. Okay, so whatever backbone that is being shown, that is shown in this pink color, and you might notice that now let me select group property and all non-polar groups. Okay, and I will only show the non-polar groups. Okay, so now the only non-polar groups are visible, and as you can see over here at this concave surface, you can see that there are a lot of non-polar residues that are exposed to solvent, right? दिख रहा है यहाँ पर? There are a lot of non-polar residues that are exposed to solvent molecule. The molecules, or you can say the residues that are colored in white now are all non-polar residues, right? And these, as you can see over here, a lot of non-polar residues are being exposed, right? So you should also know, अब यहाँ पर things become more clear, right? Check out these all non-polar side chains. They are exposed to the solvent. Okay, and you now might know by the protein physiology that all the non-polar groups are supposed to be buried inside the core of your protein, and most of the polar and charge side chain are usually exposed in your protein structure. Okay, ऐसा क्यों होता होगा? Why this must have been I mean why this might be the reason? Why non-polar side chains are supposed to be buried inside the core of your protein, and why polar side chain must be exposed on the surface of your protein?
So because polar interactions will bring the stability to protein.
Well, look, these proteins are supposed to be functioning. Proteins are supposed to be functioning at, well, say for example, in aqueous environment, right?
Yes.
The cell is mostly formed of water, okay, and when these
So
proteins are
solubility.
huh, when these proteins are expressed, they are expressed in water, right? And if the non-polar amino acid residues, the side chains of non-polar amino acid residues are exposed out in solvent, well, there would become some issue
Less
of protein
solubility.
stability, right?
Yeah.
And therefore these side chains were supposed to be buried inside the protein like this, but over here you can see that they are exposed. और दूसरी गड़बड़ क्या लग सकती है अपने प्रोटीन में कि first 15 or 16 amino acid residues, those are not those are not tightly folded. It appears that they are exposed in solvent, right? So by the end of this session, you would you would be you will find answers to these questions, okay, कि भाई ऐसा क्यों हुआ होगा, right? Good. Now let me resume. I would color the backbone again by CPK. Okay, I would show all side chains and again color them by CPK. Okay, so now everything is as normal. Okay, so do you get what are the problems as it appears in this protein?
Yes, sir.
Okay, so now, well, this is a monomer of your given protein, right? ये क्या है, सिर्फ एक सब यूनिट है आपके प्रोटीन का. Okay, so now what we will do is that we will try to color it by chains. Okay, I would select backbone and side chain and color it by chain. Okay, so when you color it by chain, when you color the model by chain, well, say many proteins are oligomeric in nature. Okay, so they are supposed to be composed of more than one protein chain. Okay, so when you color your protein by chain, this Swiss PDB viewer will assign colors, different color for different subunits. Okay, so in this way, if you color by chain, it would be a quick way to find out how many chains are there in a newly opened PDB file. So in our case, okay, in our case, we have only a single protein chain. Okay, so that would give you a cursory glance look at kind of key how many protein chains are present in your PDB file. Okay, so now what I will do is that I will go to this control panel to activate it. Okay, so if the protein has only one chain, okay, you can see that this column is empty. Okay, so this column is empty, and if you click in this empty column, you can see everything becomes red. Okay, that means you have selected the entire model now. Okay, all of the residues in your model have been selected. Okay, so now you may be aware that this lactate dehydrogenase consists of 4 identical subunits. Okay, so some PDB files, the one that we are working in, is included. Well, some PDB file, well, contains only information of unique subunits. Okay, so now they provide some some information that will enable you to generate functionally active multimeric form. Okay, so now our objective will be to build the additional subunit, and later on we will examine what is happening over their interface. Okay, so now let us redisplay the full model. Okay, reset the control color menu to backbone and side chain. Okay, and color the model by CPK. Okay, so now what I will do is that I will go to this file menu and select this one ldm.pdb file over here. Okay, it will give some general communication. Okay, I will hit this OK button. Again I will hit this OK button. I would ignore this dialog box. Okay, so when you do this, that is when you go to file and then click on this this option which is available here. So this command file and clicking, well, it gives you an idea about what are the recent files that you have opened. Okay, so this command take advantage of your Swiss PDB viewers list of recently used files. Okay, you just have to use it to open a second copy of the same file, right? So now the two models that are currently loaded over here are superimposed, so that it would appear that nothing new has been added. Okay, so you can view it in your layer infos window. Okay, as you can see that there are two copies of these lactate dehydrogenase monomers have been loaded. Okay, so now what we will do, we would repeat this procedure so that we would load 4 copies. Okay, now you see that there are 3 copies that are loaded. Okay, similarly I would hit it once more so that now there are 4 copies. I have repeated this procedure 4 times. Why? Because we want to generate a fully functional tetramer of your lactate dehydrogenase. Okay, so tetramer hai yani kya? There are going to be 4 chains. Therefore I have loaded the same molecule 4 times. Okay, do you get this?
Yes, sir.
Huh? Okay, so now this is this layer infos information, this layer information windows allows you to control the display and properties of multiple layer. Okay, so now when you do such a thing that you load multiple proteins one after another in this Swiss PDB viewer language, we call it call each and every layer that you have, each and every molecule that you have separately loaded as layer. So this is first layer, this is second layer, third layer, and fourth layer. So how many layers are there in your system now? There are 4 layers. Okay, बात समझ में आ रही है आपको?
Yes.
Yes, sir.
Okay, so now
Yes, sir.
what we will do is that we would color it by layer. Okay, so the first layer, this one is red, that means it is the one that you are visualizing over here. So the first layer is colored in red color, the second layer is colored in blue color, the third layer is colored in green color, and the fourth layer is colored again in red layer. Okay, first sorry first layer is colored in yellow, second one in blue, third one in green, and fourth
Great.
layer as red. Okay, so in this way how I have done is that I have kind of pushed the control button in your keyboard, and keeping this control button pushed, I have then pushed the tab button. Okay, control tab, control tab, control tab. In this way, you can toggle the visibility of different layer. Okay, so when you hit this control tab button, you would see that this visibility icon has moved from one layer to another layer. Okay, that shows ki which layer is visible and which layer is selected. Okay, so in this case, first layer is selected and first layer is visible. Okay, so by using this control tab buttons, you can cycle or blink between the four models. So when I use this control tab, we say that we are blinking between the models. Okay, so these models are now colored as yellow, blue, green and red. Okay, so these four models are called as layers. Okay, and on the layer infos window over here, okay, four layers are listed as 1 LDM. Now you will rename the layer. Okay, we would rename the layer so that you can easily tell apart which layer is what one. Okay, so as you can see, if we toggle by using this control tab option, okay, you can see that these layers are not really named. Okay, so what we will do is that we would rename each and every layer that is present in our protein, so that you can say that this is chain A, the first layer would be chain A, the second layer would be chain B, the third layer would be chain C, and the fourth layer would be chain D. ओके, ये ऐसा क्यों कर रहे हैं अपन? Why? Because after after generating a functionally active tetramer of your lactate dehydrogenase, it would be difficult for you to recognize what residues are present in what chain. Okay, so we would do this exercise beforehand, right? So before generating the tetramer, we would rename these layers according to their chain, so that later on it would be easier for us to identify what two chains or three chains or four chains we are dealing with. Okay, is it clear? So the next task, immediate task, would be to rename this layer. Okay, so now in your layer infos window, okay, make sure that you click over here and make the first layer as an active layer. Okay, and make this first layer as visible over here. Okay, so I have made the first layer active and made the first layer visible. Okay, so now what I will do is that this is an empty column, as you can see over here. I will click over here. Okay, and you can see that everything has been selected now. Okay, after clicking in this empty column in this control panel window, now you might have observed that everything has been have become red. That means everything, it is suggesting that all of the residues are being selected. Okay, now in order to rename this layer as chain A, what I will do is that I will go to this edit command and select this rename current layer. Okay, and over here I will give rename chain of selected group. Everything has been selected, so I will give the selected group a name of chain A. Okay, and hit the OK button, and now you can see this chain has been given the name. Okay, so this is our chain A now. Okay, this is now chain A. So now what you can do is that you can select the second layer. Okay, you can again press control tab so that now second layer is selected and visible. I would again click over here so that everything gets selected, then go to edit, rename current layer, and I would again name it as chain B. Okay, or another way around is that if you click over here, you can select the third layer. एज यू कैन सी यहाँ से भी सिलेक्ट कर सकते हैं आप. यहाँ पर अगर थर्ड ऑप्शन सिलेक्ट किया तो द थर्ड इन लेयर इन्फोज विंडो एज वेल करस्पॉन्डिंग थर्ड लेयर विल बिकम एक्टिव and visible. Okay, so again I would select the entire chain, go to edit, and rename current layer, give this layer the name chain C. Okay, again I would select the fourth layer. Okay, make it visible, select all the residues in this layer, again go to edit, rename current layer and give this name as chain D and hit OK. So now if you blink, you can see that all the layers are now renamed. This would be chain A in our functional tetramer, this would be chain B in our functional tetramer, this would be chain C in functional tetramer, and this would be chain D in our functional tetramer. Okay, so that now you can rename a part. Okay, what are the layers that are involved in your tetramer? Okay, so okay, so this is what you have done. So now what we are going to do is that we are अभी ये कर दिया अपन ने. Now it is time for us to learn how do we orient these monomer around each other so that they can form a functional tetramer. Okay, all these monomers are superimposed on over each other, right, as you can see. तो अभी नेक्स्ट टास्क क्या है? वी नीड टू रीओरिएंट ईच एंड एवरी सेपरेट मोनोमर अपॉन ईच अदर so that we would be generating a functionally active tetramer. That means what we are going to do is to fit this monomer around each other so that a functionally active tetramer can be generated. Okay, so to do so we are going to use some information that is already present in your PDB file. Okay, so what information are we going to use? I will show you this thing. Okay, so before doing this, make sure that you make the second layer from your 4 layers as an active layer and visible layer. Okay, क्या किया मैंने? I have made the second layer active and visible. Okay, make the second layer active and visible. Okay, so now when I click over this icon over here, okay, so clicking this icon, this little text icon in the main display window, this will let me consult the PDB file as text file, which is useful to have a look at the annotations as you can see over here. Now what I will do, I would scroll down a little bit where I can show you where is the crystallographic symmetry information over here, right? Okay, so if you scroll down a little bit, okay, you would end up identifying the lines that starts with the keyword matrix. Okay, so these are called as matrix line. Okay, so usually these matrix line are just located above the atom lines as such. Okay, so now in this case you can see that there are 9 matrix line. Okay, so these 9 matrix line represent 3 transformation matrices that is shown over here. So this column gives you information on this line, these 9 matrix line correspond to 3 transformation matrices. Okay, so this is the first transformation matrix, this is the second transformation matrix, and this is the third transformation matrix. Okay, and this information is enough for you to apply these non-crystallographic symmetries onto the given protein so as to generate a fully functional tetramer, or so as to fit the monomers around each other. Okay, so now again let me explain you. So this line correspond to the first line of first transformation matrix. This line correspond to the second line of second transformation matrix, and this particular line correspond to the first line of the third transformation matrix. So in this way you can identify what transformation matrix you are working on. Okay, so discussing what are transformation matrix and what are crystallographic symmetry is well way beyond the scope of our syllabus now. Okay, so for time being, if you understand that this transformation matrix can be used to apply non-crystallographic symmetry, so that it would enable you to generate the functionally active form of your protein, would be enough for you. Okay, so let us not go into more detail, let us talk about it in practical aspect. Okay, so well, now let us see how this information can be used uh, to fit the monomer around themselves. Okay, so since this second layer is active, that means we are dealing with the transformation matrix of the second monomer, that is chain B. Okay, okay, so now what I will do is that I will click on the first line of the first matrix record over here. Okay, so when I click over here, it will load the transformation matrix into a new dialog box. Okay, what I have to do is to simply click on this OK button. Okay, and when you click on the OK button, this transformation would be applied on layer B. Okay, okay, so now you can see the protein has moved a little bit. Okay, initially it was something like this. Now after applying the symmetry, it has moved a little bit like this. Okay, that means what we have done is that we have fitted the second chain or second layer over the first layer. Okay, and we would do the same thing for the remaining two layer as well. So when you select the third layer, again make it visible, it is present over here. Okay, then I will open its annotation in PDB format from using this particular icon. I will scroll down to the matrix section. Okay, and now I will click on any of the lines in the second matrix. So these three lines correspond to the second transformation matrix. Okay, so if I click over here, again the transformation box would appear, and when I click OK, the transformation would be applied for the third chain to get oriented or fitted around the already fitted two monomers. Okay, you can see the change in its transformation, something like this, right? It has moved a little bit. Okay, now I will do the same thing for the fourth layer as well. Okay, I will make the fourth layer active and visible. Again go to its annotation. Okay, annotation over here, and again go to the matrix line and click anywhere in the third matrix. I will click it over here now so that the transformation box would appear, and again click on OK so that you can see it has also moved. Okay, so now it would appear कि देखो ये रोहन सर ने क्या करके रखा है, कुछ समझ में नहीं आ रहा है. So things would be more clear if I show the visibility of each and every monomer now. Now do you see that we have fitted these monomers around each other? दिखाई दे रहा है आपको?
Yes, sir.
Okay, so after fitting this monomer appropriately by applying non-crystallographic symmetries, you have successfully generated a functional form of your lactate dehydrogenase molecule. Okay, now this is called as fitting the monomer. इसे क्या कहते हैं अपन? Fitting the monomer. And how we have fitted those monomer? By applying non-crystallographic symmetries. Okay, when I select the second layer and applied the crystallographic symmetry, that means I have fitted the second layer over first layer. Then I have selected the third layer and applied the crystallographic symmetry. इसका मतलब क्या हुआ? I have fitted this third monomer over already fitted two monomers, and finally I have selected the fourth monomer and applied the crystallographic symmetry. That means I have fitted the fourth monomer over already fitted three monomers. That is what we have done right now. Okay, is it clear? बात समझ में आ रही है आप सब लोगों को?
Yes, sir.
Okay, so now since we have done this thing, अभी आपको आपके initial सवालों के जवाब मिलना शुरू हो जाएंगे. What were the first two questions? The first two questions were कि these N-terminal amino acid residues appeared to be out there in solvent, right? Which is not the case. Why? Because when this functional tetramer assembles, well, these functional tetramer, these N-terminal amino acid residues are buried among other amino acid residues in different chain. Do you see over here? Do you see any of the exposed portion in your protein? No, everything seems to be quite compactly folded now, right? If you observe the first N-terminal amino acids, they are no longer isolated in the solvent now. Okay, and hence we answer the first question. Okay, now you might also answer the second question as to the residues that were present in the concave surface are not exposed to solvent, right? Why? Because when the subunit attains the quaternary level of organization, when they attain the quaternary level of organization, the hydrophobic side chain that seems to be exposed in the initial portion of our discussion, they are no longer exposed. Okay, they are actually deep buried inside the core of your protein. Okay, do you get this thing now?
Yes, sir.
Okay, so now the problem still is that these are all separate layers, right? ये भले ही आपको दिख रहा है कि एक साथ में फंक्शनली टेट्रामर तैयार हो गया है, बट दे आर स्टिल इन सेपरेट लेयर. Now what I will do is that I would merge all these four layers into a single layer so that you would have all the four chains at a single place, right? देखो ये अलग अलग लेयर है. These are still different four molecules, right? Loaded as four different layers. So अभी अपने को क्या करना है, ये चारों चेन्स को अपने को क्या करना है, कंबाइन करना है. Okay, so now I will make sure that all the residues in all the four layers are selected. Everything is selected in layer A, everything is selected in layer B, everything is selected in layer C, and everything is selected in layer D. Okay, make sure that everything is selected. Then what you can do is that you can go to this edit, then use this option, create merge layer from selection by layer. Okay, so now you can see that a new layer has appeared over here called as underscore merge underscore, right? So this is the functionally active tetramer now. Okay, as you can see over here, it contains all of the chains. This is chain A, then after chain A you have got chain B, after chain B you have got chain C, and after chain C you have got chain D. In this way you have fitted all the monomers around each other to generate a functionally active lactate dehydrogenase molecule. Okay, so what you can do is that you can separately save this thing. Okay, तो ये layer को select करना है. Okay, by clicking over here you can select it, or by going to this icon and then selecting over here you can select. You can do it by any either way. So make sure that everything is selected, then go to file, then go to save, then go to selected residue of current layer. Okay, and what you can do is then you can go to this building up symmetry and you can save it as one LDM tet, that give some name. Okay, and hit save button. As I have already done it, I will not do it again, right? इसे कुछ तो भी नाम दे दो टेट्रामर करके and then save it. Okay, so this is how you built up the symmetries, or this is how you fit the monomer to build a tetramer or multimers as such. Okay, and to do so, for fitting the monomer, we have applied the crystallographic symmetries, right? ठीक है, तो ये होने के बाद में आप क्या कर सकते हैं? Well, now everything is over, right? I will
Yes.
close all layers. Okay, now let us see that how these things appears. Color it by layer. Sorry, it is a single layer. Now I would have to color it by chain, right? So that you see all the 4 chains over here. तो ये होने के बाद में the next theme that we were supposed to do is that we were supposed to analyze the interaction between the interface chains as such. Okay, so what I will do is that I would first analyze what is happening over the interface of chain A and interface of chain D. Okay, दिख रहा है आपको यहाँ पर मैंने क्या किया? I have selected chain A, then I push the control button, then I select chain D, so that only chain A and D are selected while chain B and C are still not selected. Okay, now what I will do is that I will hit enter button so that I can only now visualize chain A and chain D, right? So now अभी अपने को क्या करना है? We need to figure out what is happening between the interface of these two chains, how these two chains are interacting with each other in the functionally active tetrameric form, what interactions are happening at the interface of these two chains as such. Okay, so to do so, what we will do is that I would select the group. Okay, select, and you have got a menu over here, group close to another chain. Select, select groups close to another chain, residue close to another chain. Okay, then I will select, it is asking key what should be the interface radius. I will give you the value of 5. Okay, and hit OK. Okay, maybe this is not what I have wanted. Okay. I have selected chain A. Okay, and maybe I will save this into a separate file. File, save, selected residue of current layer. Okay, and I will save it as chain AD, right? Chain AD. Okay, and hit save button. Okay, तो ये होने के बाद में क्या हो जाएगा? चेन A और D अलग से सेव हो जाएगा. Okay, so let us close this thing, and now let us save or load newly formed chain A and D. I would color it by chains. This is your chain A and this is your chain D, right? So now to see what is happening on your interface, what I will do is that I will go to this select and group close, select, and residues close to another chain. Again I would select those groups and hit OK button. Okay, and if I click this show button, now you can see only the residues that are present on the interface of around 5 Armstrong unit. Okay, so now let us prepare another view to make things quite clear. Now I would color the backbone of chain A with brown color. To do that, I would select chain, I would select the backbone option over here, select chain A, and I would color the backbone, by brown color. Okay, let me see. This is brown color. Okay, now you can see only the backbone of the chain A has been colored by brown color. Okay, similarly I would select chain D, okay, and color it in a shade of green. Color the backbone in a shade of green for chain D. Okay, now only the residues that are present in green are the backbone amino acid residues whose backbone is colored by green. Okay, now I would change my coloring selection scheme from backbone to side chain. Now whatever I would select or whatever I would color, that would only apply for backbone. Okay, so I have selected both of the chains now, because both of the chains I need to color the side chains from both of the chains now. Okay, so now I would color it by maybe type, so that the atoms are colored according to their physiological properties, okay, or physicochemical properties. So over here you can see whatever the side chains that are present in blue are basic, whatever side chains that are colored in red are acidic, and whatever side chains that are colored in yellow are polar. Okay, and non-polar amino acid residues are colored in gray. Okay, so in this way you can now identify which are the amino acid residues that are present from chain A and which of the amino acid residue are present from chain chain D. How would you do that? On the basis of the backbone color. So जो भी अमाइनो एसिड रेसिड्यू का बैकबोन ब्राउन है, that is your chain D, and जिसका भी backbone green है, that is your chain A, or it can be vice versa. Okay, so these backbone colored, you can represent what are the chains, and the side chains are colored by green. Okay, so what you can do now, what your assignment is, to to generate such a kind of view, okay, and then try to find out what are the interaction that are happening between interface, how many hydrogen bonds are present in between the two chains, how many salt bridge are present in between the two chains. Okay, now how would you define a salt bridge? Okay, I will
Interaction between an acidic and a basic
basic chain.
residue.
So now you can identify which one is basic chain. The all the amino acid residues that are colored in blue are basic. Now you would have to see that if there are any amino acid residues that are red in color, that would indicate that they are what? They are positively charged. And if they are under the distance of 4 Armstrong, that means they can putatively form a what?
Salt
Salt
bridge.
bridge. So now you need to figure out that how many, if there are any residues that whose backbone is colored in a shade of gray and side chain is colored in a shade of blue, right? And is there any corresponding residues whose side chain is colored in red but backbone is colored in green? Okay, do you get my point? So that you can identify what is happening between two chains. Okay, I will just let you know.
Yes.
Okay, if I do it like if I uh, tools compute hydrogen bond. Okay, okay, now I would focus on only hydrogen bond. I would then select the visible groups. Okay, okay, I think there is an option of displaying hydrogen bond. No, you would have to check it out for yourself. Okay, तो सबसे पहले आपको क्या करना है, check out what are the interchain hydrogen bonds. Okay, that is the hydrogen bonds that are formed between two chains. Okay, one should be from green chain and another should be from the brown chain. Okay, and the second task you need to do is that you need to figure out interchain what you are supposed to figure out interchain salt bridges, that is the interaction between two, you know, oppositely charged groups. कर सकोगे? You can use this measure button. Well, say for example, if you hit this measure button, you can find out what is the distance between the side chain. You have to make sure that the amino acid residues are in close vicinity. Okay, let me see. Let me focus on this thing now. I have figured out one. Okay, now do you see that this is one of the basic amino acid residue from chain, from a different chain which is colored in green. Okay, and over here you see blue amino acid residue which is present in this brown chain. Okay, so potentially these two chains can form a disulfide bridge, right? So how do you find out the distance now? We find out the distance by using this measure menu. Okay, click on one atom and click on the corresponding atom. तो कितना डिस्टेंस है इन दोनों दो साइड चेन्स के बीच में? इट इज अबाउट 4, राइट? If I hit escape, you come out of that function. Okay, so this is one possible, right? So this can rotate, this can also rotate. Okay, and it can also form interaction between these two atoms as well. तो देखो, if this particular atom can form, you know, salt bridge with this particular side chain. Okay, so in this way this acidic residue from one chain can form a salt bridge with this particular residue in another chain, that is different chain in your interface. Okay, so this is how you can do it, right? And you can identify what are the two residues using this button. Okay, so what is this residue? It is glutamate 258 is forming a salt bridge with histidine 73. That is how you can identify. ओके, बात समझ में आ रही, क्या करना है?
Yes, sir.
Okay, so here is how we end up with our discussion on fitting the monomers, right? And subsequently analyzing the interface between the fitted monomers as such. Okay, so what is your objective now? What is your assignment now? To figure out how many hydrogen bonding partners are present in between the two chains. Okay, chain A and chain D. If there are any salt bridges that are stabilizing the two chains. Okay, and you can prepare this such a view, and you can save this view file, save current layer. Okay, then you can have like view AD, right? And then you can save it. Okay, I have saved it in some different location. View AD, right? Maybe I will load it in some different instance. Okay, you can save this view, right? And you can analyze it by your own pace, right? Look, I have identified another disulfide bridge. Okay, I would rotate it. Now check out that you have this amino acid residue. What is it? Glutamate 258 from, say, green chain, right? And you have got this arginine 266 from, okay, they are present from the same chain. We need to figure out someone from, that is one and the same thing that we have already done, I suppose. Okay, these things can also be in salt bridging, right? You see this amino acid residue, what is it? Lysine 262 from brown chain, right? And you have got this aspartate 43 from green chain. Okay, so we can focus our view and see if they form a salt bridge. I would click it over here and click it over here. Yes, there is another salt bridge over here, and this salt bridge is between the lysine of 262 from one chain and aspartate 43 from another chain. Look, I have identified you already two salt bridges. You are supposed to now check if there are many more. Okay, and list it out. Maybe you can draw, or you can save this screenshot, right?
Yes.
Okay, so this is how you do structural analysis and protein modeling. Okay, in our next lecture we would be talking about protein structure prediction. Till now we were talking about protein modeling and what you can do by analyzing the existing protein models. But now situation might arise that you might be interested in certain protein but whose structure might not be deduced by experimental techniques. The models might not be present in PDB. In such case, you can predict the structure of your interested protein molecule. That is what would be the focus of discussion of our next subsequent lectures. Okay,
Okay. Yes.
ठीक है. So सबको समझ में आ गया क्या करना है? Okay, you can
Yes.
view this video a bit later. Again, try to do the same thing that I have done, understand it what we have done, and do your assignment appropriately.
Okay.
Okay, you need to figure out interchain hydrogen bonds, and you need to figure out interchain what?
Dye. Salt bridges.
Salt bridges. Okay, so you can identify hydrophobic interaction as well. Hydrophobic interaction kaise hoge? Polar group from one chain, the side chain of polar group from one chain forms a cluster of the side chain of hydrophobic side chain of another chain, as you can see over here. This is a hydrophobic patch, or over here you can see a hydrophobic interaction between the two chains, right? This is hydrophobic interaction,
Yes.
right? तो क्या करना है? You have to do three things. First, interchain hydrogen bonds, interchain interchain क्या?
Salt bridges.
Salt bridges, and third,
Hydrophobic
interchain
interactions.
hydrophobic interaction. Okay,
Yes.
चलो ठीक है. I hope you guys understood and enjoyed today's session, right? So if there are any
Yes.
question, please shoot out. And if there aren't any, maybe I will stop the recording now.